Hello all, welcome to Paritranaya. In this class, we shall discuss about mitochondrial replacement therapy, a very important topic from the exam point of view. So let us understand what is this mitochondrial replacement therapy and why it is required. But before that, we also need to understand what is genes and what is inheritance, and based upon that, we will be able to connect to this topic. So we all know that every human cell, it consists of multiple things. Okay, it consists of multiple things. For example, it may have a nucleus, it may have ribosomes, mitochondria, centrosome, microfilament, many things. But here we are interested in two things when it comes to cell. One is nucleus, another one is mitochondria. So why these two things are important for this discussion? Because they consist of genes. So nucleus also consists of genes. Similarly, mitochondria also consists of genes. Now we often hear, when we look at someone, we say that, so you look similar to your father, you resemble your father, your eyes resemble your mother. Similarly, we also say that, okay, so as his father was facing with the problem of heart diseases, similarly his son is also facing with the heart related diseases. So all these things we are able to infer because we know that there is an inheritance. The child, it has inherited some of the physical features from its parents and also characteristics from its parents and also diseases as well. So to understand these things, it becomes important for us to understand about the genes. So all these characteristics which a child inherits, it inherits from its parents. So when it is inheriting, it will inherit either from the nucleus or it will inherit from the mitochondria. So a child inherits the DNA or the genes from the parents, either from the nucleus or the mitochondria. So here one important thing to observe is the nucleus consists of majority of the genes. For example, 99.95% of the genes are present in nucleus and very minuscule. So 0.05% of the genes is present in mitochondria. So a person's physical characteristics or other characteristics or behavioral characteristics, so majority of them, so they are inherited from the genes which are present in nucleus. Okay, so this is a very important point to note. Now next we need to discuss about mitochondrial replacement therapy. So what is the connection between this cell and mitochondrial replacement therapy? Now this is an example which I have taken. So here I assume that this is the cell of a father, and especially it's the what kind of cell? It's a sperm cell. It's a sperm cell. And here I have assumed it to be a cell of mother, and I assumed it to be what? Egg cell. I assumed it to be egg cell. Now this is the cell of child. Cell of child. Cell of child. Now in father we have nucleus, we have mitochondria. In mother we have nucleus, we have mitochondria. Child has nucleus, child has mitochondria. So when it comes to inheritance, the genes which are present in this nucleus, so they have been inherited from both father as well as mother. So while inheriting, so there will be a mixture. It means some of the genes will be inherited from the father and some of the genes will be inherited from mother. That's why few of them, their eyes resemble their mother and their nose resemble their father. Okay, so one is prone to heart disease because his father is suffering from heart disease, and another one may be prone to diabetes because he has inherited the genes of his mother who is a diabetic. So this DNA or this nuclear DNA, so it consists of material which has been inherited from both father as well as mother. An important point to note. So I already mentioned that mitochondria also consists of genes. But this mitochondrial genes, a child does not inherit from father. A mitochondrial genes, a child does not inherit from father, and completely it is inherited from mother. This is a very important point to note. When it comes to nuclear DNA, it has been inherited by both father as well as mother. But when it comes to mitochondrial DNA or genes, a child has inherited only from mother. Okay, only from mother. Now here, if the mother's mitochondria has some kind of defects, some kind of defects. So because of the defects, mother is facing some kind of she's a victim of disease, some diseases. Okay. So the same disease, it should not be passed to the offspring, to the child. So to prevent that, so there is a technique to prevent the disease from being passed from the mother to the child, especially the disease related to mitochondria. There is a technique called as mitochondrial replacement therapy. So here, the unhealthy mitochondria will be removed, and the healthy mitochondria of another mother will be brought in, so that when a child is born, so majority of the characteristics, it will be inheriting from what father as well as the actual mother, but mitochondrial related characteristics are very minuscule, so it will be inheriting from another mother. Okay, so this technique is called as mitochondrial replacement therapy. So now next we shall discuss in detail about the various mitochondrial replacement therapies that exist. These topics are very important from exam point of view. So as I mentioned, it's a groundbreaking technique that uses genetic material from 3 different people. Who are the 3 different people? One is the actual father and mother, and the third person is the person who have donated the cell with good mitochondria, good mitochondria, or correct mitochondria with no faults. So why it is used? To prevent certain inherited diseases, genetic diseases that cannot be treated. Until now, there is no treatment for such kind of diseases. So we want to prevent them from being passed from mother to offspring. Okay, so this is a very important point. So mitochondrial replacement therapy is used to prevent a disease being passed from mother to offspring, especially mitochondria related diseases. So here in this technique we use IVF procedure. So already we have discussed about the IVF procedure, so you can visit our video related to it. So here X defective mitochondrial DNA is replaced with healthy DNA from a female donor. So how it exactly happens in techniques we shall discuss later on. So in this context, as a cell consists of multiple entities like nucleus, mitochondria and many other things, now we'll be focusing on only mitochondria. So mitochondria, it is a powerhouse of the cell. So all the energy which the cell receives, it comes from mitochondria. This is also important. So as I mentioned, mitochondrial DNA is very, very minuscule, 0.054%, whereas the majority of the DNA is present in nucleus. Okay, so nuclear DNA is very important in determining the personal characteristics and traits. So while a child's DNA is a mixture from both the mother and the father, mitochondria are separate packages of genetics that come solely from mother. This also we have discussed. So in case of mitochondria, it is completely inherited from mother. Some people have mitochondrial diseases, a problem with the genetics in their mitochondria, which can be severe life-threatening conditions, although it is very rare. So mitochondrial diseases, they are very rare, but sometimes if it occurs, then it will be life-threatening. So in order to prevent these life-threatening diseases related to mitochondria from being passed from mother to child, so we have these MRT techniques. So the common techniques in use are pronuclear transfer, maternal spindle transfer, polar body transfer. So all these techniques are very important from exam point of view. So let us look into how these techniques work. So first we shall discuss about pronuclear transfer. Pronuclear transfer. So here I have taken 2 couples. So patient couple and donor couple. So let me call them as A and B. So B is female here. Next, let me consider X and Y. So Y is female. Y is female. Now the couple who wants to have children. So in this couple, B is suffering from some kind of mitochondrial related diseases. Whereas F is not facing any is not having any kind of mitochondria related diseases. So now what we want to do is, so we want to take the healthy mitochondria from F. That procedure we shall see. So this is a fertilized egg from patient with a faulty mitochondria. So here A is sperm and B is egg. So both of them, they have been mixed together and it has been fertilized. So when it has been fertilized, now this is a single egg. Sorry, this is a single cell of the next embryo which is going to develop. So here, if you look at nucleus, so this nucleus, it consists of characteristics of both father as well as mother. Now here the entities which have been represented, there are various entities. I'll just focus on mitochondria, but this mitochondria is faulty. So what we'll do is, so we'll leave this mitochondria and entire cell, only we'll take out the nucleus. Okay, we'll take out the nucleus. So here this nucleus, it is healthy and it consists of characteristics of both A as well as B. Next here, similarly, we'll take the fertilized egg of what? X and Y. So here the sperm of X and egg of Y, so they have been fertilized. So the resulting cell of the zygote. Okay, zygote. Now what happens here? So this nucleus, it consists of characteristics from X and Y, because it has inherited the characteristics from X and Y. So we are not interested here. So what we'll do, we'll remove this. But the mitochondria which is present over here, which belongs to mother Y, it is healthy. So we want this mitochondria. So in the next step, here we have healthy mitochondria which has been received from here, and in this empty box, because this has been removed here, nucleus. So here this nucleus will introduce. So this nucleus is healthy. Okay. Now what we are seeing, except nucleus, the other things belong to this pair, whereas nucleus belongs to A and B pair. So it means the resulting child, it will be inheriting the characteristics of A and B. Okay, A and B, a very minuscule 0.05%, it will be inheriting from F. So predominantly its characteristics will be influenced by A and B. Okay, at the same time, the child will not have any kind of mitochondria related diseases. So next, this cell divides, it becomes embryo, and subsequently it attaches to the womb and it starts developing and the pregnancy occurs, and the resulting the newborn child will not have any kind of mitochondrial diseases. So this technique is called as pronuclear transfer technique. Okay, so this is one of the mitochondrial replacement therapy. An important point to note here is, here replacement of mitochondria, it took place after the fertilization. Fertilization took place first, after that replacement of mitochondria took place. Now the next technique that we will be discussing, there first replacement will take place, next fertilization will take place. Okay, so this is called as maternal spindle transfer. So here, okay, so mother B, so donor Y. So mother B has unhealthy mitochondria but good nucleus. So here donor Y, they have healthy mitochondria. Okay, healthy mitochondria. So mother's egg, so donor's egg. Now from the mother's egg, what we will do, we will just remove out what? Nucleus. And from donor, what we will do, donor's egg, we will remove the nucleus. Okay, and we will make it blank. So here in this blank, what we'll do, we'll come and add it. So this is the case. So here the mitochondria, healthy mitochondria, we have received from Y, whereas this nucleus has come from B. So it means this egg cell has characteristics predominantly which have been inherited from B. And at the same time, it does not have defective mitochondria. Now this egg cell will be fertilized with sperm of A, that is B's husband. So now the resulting child will have characteristics that have been inherited from both A and B, and very minuscule characteristics as part of the mitochondria that have been inherited from Y. Okay, so again, this is another technique of preventing mitochondria related diseases from being passed from mother to child. So an important observation here is, we have replaced the mitochondria before the fertilization itself. So before fertilization occurred, so we have replaced mitochondria, whereas in the previous technique, that is pronuclear transfer, we saw that mitochondrial replacement took place after the fertilization. So from the exam point of view, at least we need to have this superficial level information. Sometimes that information will be sufficient to tackle what any questions. Okay. Next, similarly, we have one polar body genome transfer. So here we shall just understand the superficial level. So what is polar body? A cell with very little cytoplasm that is created when an egg cell divides. So generally when an egg cell divides, so there are two cells formed. So this cell, it consists of little cytoplasm. So let us not go into the deeper aspects. A cell which consists of little cytoplasm, so that is called as polar body. So instead of just using the nucleus here, okay, the polar body will be used. Polar body will be used. So this much information, if we are able to retain, that would be sufficient. So here we shall understand that. So polar body genome transfer is one of the techniques used in mitochondrial replacement therapy. So this can be used as part of maternal spindle transfer or pronuclear transfer. So again, this is important. Apart from these, there is also one more technique called as cytoplasmic transfer. So it is one of the old technique. It is no longer in use because of concerns. So important. So now they will not ask us in detail about the cytoplasmic transfer, but at a superficial level they may ask. So cytoplasmic transfer technique is used in what? So it is used in mitochondrial replacement therapy. So the various discussions that we have made, so they are very much important, especially from the prelims point of view. So UPSC often focuses on its applications when it comes to science and technology. So from that perspective and looking at the pattern of the UPSC, so the concept which we have discussed is very important. Similarly, in the forthcoming classes, we will be discussing about the other important science and technology related topics from the exam point of view. For all our further updates, subscribe to our channel and hit on the bell icon. Thank you.